So as we have seen earlier, uh, in the water absorption, the kind that initially enters the large intestine is isotonic. However, in the colon, more water than the electrolyte is absorbed, and the fluid becomes hypertonic. Okay, leading to water being absorbed against the concentration gradient. Okay, the process of water absorption is controlled by the sodium potassium ATPase, located by the basolateral and the lateral membranes of the epithelial cells. The sodium pumped into the extracellular space and tight junction at the luminal membrane. Okay, so the sodium is pumped into the extracellular space and tight junction. Okay, uh, at the luminal membrane, prevent the diffusion of the sodium and chloride back from the extracellular space into the lumen. This leaves the hypertonic solution close to the lumen, causing the water to flow by the osmosis. Okay, the electrolytes are absorbed by the variety of mechanism. The electrolytes are absorbed by the variety of mechanism similar to those of the small intestine. Okay, this essentially a net movement of potassium and bicarb into the lumen of the large intestine. With essentially the net movement of the potassium and bicarb into the lumen of the large intestine, because the potential difference set up by the asymmetrical absorption of sodium and chloride across the epithelium. So the sodium and chloride from the lumen, sodium and chloride uh, will come into the bloodstream, and the potassium and the bicarb will excreted into the lumen. Okay, now intestinal flora. The commensal intestinal bacterial flora have a role in keeping. Okay, the intestinal flora, the commercial intestinal bacterial flora have a role in keeping the pathogenic bacteria at bay by competing for space and nutrient. Okay, uh, the intestinal flora keep the pathogenic bacteria at bay by competing for space and the nutrient, converting conjugated bilirubin to the unconjugated bilirubin, some of which is reabsorbed and the excreted into the urine. Okay, the conjugated bilirubin will converted into the unconjugated bilirubin and reabsorbed and excreted into the urine, and stercobilinogen, which is excreted in the feces. Okay, this conversion is done by intestinal flora. Okay, the synthesis of vitamin K, B12, thiamine, and the riboflavin, synthesis of the vitamin K, B12, riboflavin, and thiamine will also occur in the intestinal by the intestinal flora. Then the breakdown of primary bile acid to the secondary bile acid, and breakdown of the cholesterol, some food additives, and the drugs also occur by the intestinal flora. Okay, it cause keep the pathogenic bacteria at the bay, then converting the conjugated bilirubin into the unconjugated bilirubin, which is reabsorbed and secreted into the urine, and convert into the stercobilinogen, which excreted into the feces. Then the synthesis of the vitamin K, B12, thiamine, and the riboflavin, and the breakdown of the primary bile acid into the secondary bile acid, and breakdown of the cholesterol, some food additives, and the drugs. It's also done by the bacterial flora. Then, what is the function of the liver? So, functional anatomy. The liver consists of four lobes, which made up of hexagonal lobe. Okay, and functional unit of the liver. Liver consists of the four lobe, which made up of hexagonal lobules, which is the functional unit of the liver. Each lobule consists of a central vein that eventually become part of the hepatic vein. Surrounding the central vein are single columns of hepatocyte relating outward. Between the hepatocyte are some canaliculi which drain into the bile duct on periphery of the lobule. At each of the six corner of the lobule lies a portal triad, okay, comprising branches of the hepatic artery, the portal vein, and the bile duct. Okay, so this is the hexagonal lobule, which is functional unit of the liver. Okay, each lobule consists of central vein, central uh, interlobular or interlobular vein, which part of the hepatic vein. Okay, surrounding the central vein. Okay, surrounding the central vein are a single column of hepatocyte radiating outward. Between the hepatocytes, okay, this is the hepatocyte. Between the hepatocytes are small canaliculi, okay, are small canaliculi which drain the bile duct on the periphery of the lobule. Small canaliculi which drain the bile duct to the periphery of the lobule. Now, at each of the six corners of the lobule lie a portal triad. There is the portal triad, a portal vein, portal artery, and the bile duct. Okay, uh, comprising branches of the hepatic artery. Okay, there is the branches of the hepatic artery. And the portal vein and the bile duct. Okay, so the portal vein, portal artery, and the bile duct. Function. So the main function of the liver are fetal hemoptysis, fetal hemoptysis, hematopoiesis, sorry, fetal hematopoiesis, then the protein metabolism, fat metabolism, carbohydrate metabolism, bile production, storage of vitamin and minerals, bone metabolism in form of hydroxylation of the vitamin D. Okay, then production by filtration of portal blood production, sorry, protection by filtration of portal blood, and phagocytosis of bacteria, antigen by Kupffer cell. Okay, so what we have seen in the function, that the main function of the liver are the fetal hematopoiesis. Okay, then the protein metabolism. In the protein metabolism, the synthesis and the secretion of the albumin, synthesis and secretion of the albumin, synthesis of plasma protein, formation of urea from the ammonia, deamination of the amino acid, synthesis of the coagulation factors, and metabolism of the polypeptide hormone. Okay, synthesis and secretion of the albumin, synthesis of plasma protein, synthesis and secretion of the albumin, then the plasma protein, then formation of the urea from the ammonia, then deamination of the amino acid, and synthesis of coagulation factor. Okay, synthesis of the coagulation factor and metabolism of the polypeptide hormone. Okay, metabolism of the polypeptide hormone. Then the fat metabolism, formation of the lipoprotein and fatty acid, formation of the lipoprotein and fatty acid, synthesis of the cholesterol, conversion of the cholesterol to bile salt, and conversion of the carbohydrates and protein to fat, ketogenesis, metabolism and excretion of the steroid hormone. Okay, then carbohydrate metabolism, so the gluconeogenesis, glycogenesis, and the glycogenolysis. Bile production, so the synthesis of bile salt, production of the alkaline fluid for neutralization of gastric acid in the intestine, excretion of the waste metabolites, and detoxified substances from the body in the bile. Okay, then the storage of vitamins and minerals, folate, B12, fat soluble vitamins, iron, and copper. So what we have seen here, that the in the protein metabolism, synthesis of the secretion of the albumin, synthesis of the plasma protein, formation of the urea from the ammonia. Okay, then the deamination of the amino acid, synthesis of the coagulation factor, and the metabolism of the polypeptide hormone. These are so many things which are done in the, which are happens in the liver. Okay, then the fat metabolism, formation of the lipoprotein, fatty acid, cholesterol, then conversion of the cholesterol to bile salt, conversion of the carbohydrate and protein to fat, ketogenesis. Okay, then reabsorb, so that metabolism and excretion of the steroid hormone. Then carbohydrate metabolism, gluconeogenesis, glycogenesis, and the glycogenolysis. It also occur there. Then the bile production, synthesis of bile salt, production of alkaline fluid for neutralization of the gastric acid in the intestine. Okay, production of the alkaline fluid, bile salt, alkaline fluid, then the excretion of the waste metabolites and detoxified substances from the body in the bile. Then storage of vitamins and minerals, so folate, B12, fat soluble vitamins, iron, and copper. Okay, they are stored here. Then bone metabolism in the form of hydroxylation of the vitamin D. Okay, then protection by filtration of portal blood. Okay, protection by filtration of portal blood, and the phagocytosis of bacteria and antigens by Kupffer cells. Okay, Kupffer cells cause the phagocytosis of the bacteria and the antigens. Now we'll see the function of the small intestine. So uh, the small intestine is the main site for digestion and the absorption processes. Okay, is the main site for the digestive and the digestion and the absorption process occur in the small intestine. Okay, mostly of the small intestine via the peristalsis and segmental contraction facilitates mixing of intestinal content, the bringing of intestinal content into the contact with the absorptive intestinal brush border, and the forward propulsion of the contents. The small intestine absorbs water, electrolyte, carbohydrate, amino acid, mineral, fat, and the vitamins. Okay, so what we have seen, that the small intestine is the main site for the digestion and the absorption process. Motility of the small intestine via the peristalsis and the segmental contraction facilitate the mixing of the intestinal contents, mixing of the content, then the bringing of the intestinal content to into contact with the absorptive intestinal brush border. Okay, bring the this content to the absorptive intestinal brush border. Okay, and the forward of propulsion of the contents. Okay, the small intestine absorbs water, electrolyte. Okay, small intestine will absorb the water, electrolyte, carbohydrate, amino acid, minerals, fat, and the vitamin. Okay, amino acid, mineral, fat, vitamin, electrolyte, and the water, and the carbohydrate. Okay, then duodenal chyme. So, duodenal chyme, the chyme that first enters the duodenum, is acidic. Okay, the kind that first entered the duodenum is the acidic hypertonic and only partially digested. There is an osmotic movement of water across the freely permeable wall, which leads to the content becoming isotonic and addition of both bicarb from the pancreas and the bile from the liver, which neutralize the acidity, allowing further digestion of the kind performed by the addition of the enzyme from the pancreas, liver, and the intestine itself. Okay, that's the duodenal chyme. Then functional anatomy. The mucosa of the small intestine has a much greater surface area for absorption than other part of the gastrointestinal tract. Okay, in the small intestine, the mucosa has a much greater surface for the absorption. Factors increasing the surface area, including the small intestine, is very long, about 5 meter in length. Then the inner wall of the small intestine is covered by the numerous fold of the mucous membrane called the plica circularis. Okay, that's called the plica circularis. The inner wall, okay, the lining of the small intestine is folded into many finger-like projections called villi. Okay, the inner wall of the small intestine is covered by the numerous fold of the mucous membrane called the plica circularis. Then the lining of the small intestine is folded into many finger-like projections. Lining of the small intestine folded into the finger-like projections called the villi. Okay, then the surface of the villi is covered with a layer of epithelial cell. Okay, the surface of the villi is covered by the layer of the epithelial cell. Okay, surface of the villi is covered by the layer of epithelial cells, which in turn have many small projections called the microvilli. Okay, the surface has epithelial cells which in turn have many small projections called the microvilli that projects towards the lumen. Okay, that projects towards the lumen of the intestine, forming the brush border. Okay, the microvilli are covered by a glycocalyx. Microvilli are covered by the glycocalyx. Okay, which contain many enzymes which are involved in digestion. Okay, this microvilli contain. Okay, so what was that? The surface of the villi is covered by the layer of epithelial cells which have many small projections called the microvilli that project toward the lumen of the intestine. Okay, forming the brush border, and it will form the brush border. This microvilli covered by the glycocalyx which contain many enzymes, glycocalyx. Okay, that contain many enzymes which are involved in the digestion and the transport. Okay, this enzyme glycocalyx contain many enzymes which are involved in the digestion and the transport. Okay, which each villus contain a single blind-ended lymphatic vessel. Okay, each villi contain a single blind-ended lymphatic vessel called the lacteal, and also a capillary network via which nutrients are absorbed. Okay, this microvilli covered by the glycocalyx which contains many enzymes which are involved in the digestion and the transport. Each villus contain a single blind-ended lymphatic vessel called the lacteal. Okay, and also a capillary network via which the nutrients are absorbed. Okay, so this is the lymphatic vessel. Okay, this is the muscularis mucosa. Okay, and this is the duodenal gland. Okay, these are the blood sub vessels, and this is the lacteal. Okay, this is the villi. Okay, absorptive centers. These are the capillary, artery, and Now goblet cell, intestinal crypt, microvilli. Okay, here there is the microvilli. Okay, this lacteal is located in the villus. Okay, and this is the microvilli. Microvilli, this is the brush border. Water absorption and the sodium pump. As the content of the intestine are isotonic with body fluids and mostly have the same concentration of the major electrolyte. Okay, to the body fluid and have the same concentration of the major electrolyte. Their absorption is active. Water cannot be moved directly but follows the osmotic gradient set up by the transport of ions primarily mediated by the sodium pump. Okay, set up by the transport of the ion primarily mediated by the sodium pump. Absorption is active. Water cannot be moved directly. Okay, this water cannot be moved directly but follow osmotic gradients. Okay, it follow the osmotic gradient set up by transport of the ion. Okay, primarily mediated by the sodium pump, set up by the transport of the ion, primarily mediated by the sodium pump. Water cannot be moved directly but follow the osmotic gradient set up transport of the ion primarily by the sodium pump. Okay, sodium potassium ATPase are located on the basolateral membrane of the epithelial cell pump. Three sodium ion from the cell is exchanged for the two potassium ion against their respective concentration gradient. This leads to low intracellular concentration of the sodium and the high intracellular concentration of the potassium. The low intracellular concentration of the sodium ensures the movement of the sodium from the intestinal contents into the cell down its concentration gradient by both the membrane channel and transporter protein mechanism. Sodium is then rapidly pumped again by the basolateral sodium pump. Potassium leaves the cell across the basolateral membrane down into the concentration gradient linked to an upward movement of chloride against the concentration gradient. Having entered the cell across the luminal membrane down its concentration gradient. This movement set up an isotonic gradient between the lumen and the blood, leading to water absorption following the movement of the sodium and chloride across the luminal membrane. Okay, up to 9 liter of the water is absorbed from the GI tract per day. Most of form from the small intestine, especially the jejunum. Okay, so into the jejunum this water is absorbed, and also the sodium potassium ATP is located in the basolateral membrane of the epithelial cell, pumps three sodium ion from the cell in exchange from to potassium ion against their respective concentration gradient. This leads to low intracellular concentration of the sodium and a high intracellular concentration of the potassium. Low intracellular sodium ensures a movement of the sodium from the intestinal content into the cell down in concentration gradient by both the membrane channels and transporter protein mechanism. Sodium is then rapidly pumped again by the basolateral sodium pump. Potassium levels leaves the cell across the basolateral membrane down its concentration gradient, linked to an outward movement of the chloride against its concentration gradient. Having entered the cell across the luminal uh, membrane down its concentration gradient. Okay. Now, function of the stomach. So, the stomach receives food and fluid from the esophagus. Okay, stomach receives food and fluid from the esophagus, mixes it with the digestive juices to form chyme. Okay, food and fluid from the esophagus, mixes it with the digestive juice to form the chyme and release its content into the duodenum. The stomach acts as a reservoir for food as it is very distensible. When empty, it has a volume of about 50 ml, but it has capacity of up to 3 to 4 ml. Okay, there there are two sphincters at each of the gastric orifice. The lower esophageal sphincter protect the esophagus from reflux of the gastric content, and the pyloric sphincter control the flow of gastric content into the duodenum. Okay, what is the function? The main function of the stomach are store food temporarily, as it can be ingested more rapidly than it can be digested. Okay, the ingestion store food temporarily as it can be ingested more rapidly, than it can be digested. Okay, chemically and mechanically digest food using acid, enzyme, and the uh, kerning movement. Okay, acid, enzyme, and the kerning movement. Digest food by using the acid, enzyme, and the kerning movement. Regulate the release of resulting chyme into the small intestine. Regulate the release of resulting chyme into the small intestine. Okay, secrete intrinsic factor which is essential for the absorption of the vitamin B12. Okay, so it regulate release of resulting chyme into the small intestine, and secrete intrinsic factor which is essential for the absorption of the vitamin B12.